Good morning, everyone. Welcome to our monthly EU OPENSCREEN webinar, and thank you for taking the time to be here with us today. I'm Dr. Kathis Kopelitou, and I will be your host for today's webinar. Today, we have the great pleasure to have here with us today Dr. Patrick Aloy, who is a research professor in the Catalan Institute for Research and Advanced Studies and principal investigator of the Structural Bioinformatics and Network Biology Lab and the Institute of Research in Biomedicine in Barcelona. For 25 years, Dr. Aloy has been developing and implementing new technologies and algorithms, applying state-of-the-art methods to specific problems, and bridging the gap between theoretical models and experiments in different disciplines. The main goal of his lab is to combine molecular, cell, and computational biology to unveil the basic wiring architecture and dynamics of physiopathological pathways to increase our understanding of how biological systems change from the healthy state to disease. In the last years, he has been developing resources to process, harmonize, and integrate bioactivity data on small molecules, providing compound bioactivity descriptors that push the similarity principle beyond chemical properties. Currently, the main research line in the lab is to collect heterogeneous data sets and develop novel methodologies to integrate different layers of regulation to unveil disease signatures. Today, we are going to discuss how blending chemistry and biology can enable systems pharmacology. Patrick mentions that big data analytical techniques and artificial intelligence have the potential to transform drug discovery as they are reshaping other areas of research, of science and technology, but we need to blend biology and chemistry in a format that is amenable for modern machine learning. Before I give the speech to Patrick, I will kindly remind you that this webinar is being recorded. And at the end of the webinar, we will be running a live Q&A session where you can ask your question directly to our speaker or raise your hand to comment. So Patrick, I now will give the speech to you. I will stop sharing my screen now, and we will be happy to hear your presentation.
Well, thank you very much. Good morning, everybody, and thanks for the invitation and the introduction. Let me share my screen. Can you see it fine?
Yes.
Okay.
And maybe, yep, you can, yeah, we can see now the full mode. Yeah, great. Thank you, Patrick.
Okay, perfect. So basically, this is a bit the rationale of everything that we are doing in the lab, and that's that, as you well know, biology is a very complex system. So this means that by studying the isolated parts of this system, we cannot figure out the complex behavior that it will have, right? And these are just a few examples. So this protein-protein interaction network related to Alzheimer's disease that we charted in the lab a few years ago. So every time you see a green dot here, this is the protein, and two dots, two proteins are connected if these proteins do physically interact. So this is a well-known metabolic network with a Krebs cycle, etc. You're all well used to see these sort of things. And while these are clearly complex systems, these are things that happen within one cell. This happens in neurons, this happens in any cell. But of course, if we're talking about human biology or human biomedicine here, the complexity doesn't stop in the cell, because we know that cells do interact to form tissues, tissues do interact to form organs, and finally organs do interact to form the whole human body. And as you're well aware, most of the first-in-class drugs are still discovered using phenotypic screens. But it seems that the preferred methodology by most of us is the target-directed screen. So this means that what we do is we take one of these yellow spots in here, one protein, we isolate it, we put it in a test tube, and then we try all our screens, all our compounds on this particular target. This is great. Of course, it's much easier to work with one protein than to work with the whole system. But this also means that then when we try to put this drug back into the complexity of the system, the more complex the model that we try, the more chances we have that this drug is not going to do the effect that we want it to do. right? So basically, the main aim of my laboratory is to see whether we can somehow capture this complexity and introduce it in the first steps of the drug discovery proceeding, right? And of course, we're in 2023. It means that we're doing this through the use of big data and artificial intelligence, because we hope that it can capture the emergent properties of the system. But then the big question here is, how can we integrate these complex properties of the biological systems in these first steps of drug discovery? So this equation has two different elements, right? So this is the biology and there's the chemistry, and both walls are pretty complex. So what we think we have to do is to try to capture this complexity of both walls in a form that computers understand, so that we can play again with these nice tools that our colleagues in computer sciences, artificial intelligence are developing. So, but we need, as I said, to format all this information in a way that the computers understand. And this is basically descriptors in the form of vectors. Chemistry first. So you see a chemical molecule, you're used to see this sort of representations, but unless you know exactly what this does, which is Klebeck, so it's pretty difficult that you can sort of guess the function only from its molecular structure. So what we and others have been doing for the last 30 years, so this is the basics of chemoinformatics, is again try to capture the chemical structure of all these compounds in the form of a vector that then we can compare, right? So we can do this with Morgan fingerprints, with the scaffolds, the structural keys, you name it. And the key here is what we call the similarity principle. So we have known for many years now that whenever two molecules have similar structures, there is a tendency that these two guys will have similar bioactivities when exposed into a biological system. What we have discovered in the last 10 years or so is that not only molecules with similar structures tend to have similar bioactivities, but also those molecules that trigger similar transcriptional responses when exposed to cell lines tend to have similar bioactivities, or at the highest level, drugs that trigger the same side effect profiles also tend to have similar bioactivities, or those compounds that kill certain cancer cell lines also tend to share bioactivities. So what we started to do already 7 years ago in the lab is saying, okay, so can we encode, can we capture the bioactivity of these small molecules in the same way that we capture their chemical structure, so that we can transform not the chemical structure of these compounds in a vector, but the effects that these different molecules will do when they're exposed into a biological system. And basically what we did is to create a tool, a resource that we call the chemical checker for obvious reasons. I mean, it has this checkerboard format that is composed of about 1 million bioactive molecules for which we have experimental information, right? And then we have divided this chemical checker into 5 different levels of complexity that follow a bit the way we understand drug action. So in the simplest level, of course, we have the chemistry. We have different ways of describing the chemical structure of these compounds, to the fingerprints, the scaffold, structural keys, etc. Then for a small molecule to be bioactive, it means that at some point it has to bind to a biomolecular target. This is the second level of complexity. And here we have ways of describing the mechanism of action, the genes that will metabolize these small molecules, binding effects, etc. Then when you modulate the action of one protein, this modulation gets spread out, this signal gets spread out in the cell through different types of networks. So here we have metabolic networks, we have signaling pathways, we have the interaction protein-protein interaction networks, etc. And all these are effects that globally we can capture in cell-based assays of, again, different sorts. So we can measure differential gene expression, or we can have huge cell line panels and see which ones survive, which ones die in front of a certain chemical. We have chemical genetics, we have high content screens. So you put your compound cell line, look under the microscope, and you measure more than 1,000 different morphological features. And finally, for the few compounds that reach the clinics, this is the maximum level of complexity. We have information about the therapeutic areas, the therapeutic indications, side effects, toxicology, etc. So big numbers are very powerful, but are usually very difficult to interpret. And I'm just going to give you a glimpse of two cases in here so that you can capture, you can see the immense reduction in complexity that we are doing in the chemical checker, right? So for the transcriptomics effect, so here these descriptors try to capture in only one vector the transcriptional effect that one compound does when exposed to a cell lines. But basically, this information comes from the LINX 1000 repository in the Broad Institute. And here we have information about 20,000 molecules. But then these 20,000 molecules have been tested between 3 and almost 80 different cell lines at 3 different concentrations, and the effects have been measured at 3 different time points, right? And we're trying to condense all this information in the form of one single descriptor. So if we look at the drug response, so here we are comparing different measurements of drug response of 15 different drugs measured by different labs. You can see that apparently only one of these drugs have comparable or correlated measurements. But this is not exactly this. When you process this data as you should, you can see that the correlation is indeed pretty high, right? So, and this slide just captures in this one picture about 3 years of work in my laboratory, right? So all this is published, so you can go and check it out. I'm not going to bore you with the details, but it's basically each one of these spaces has a huge amount of processing and harmonizing of this small molecule related bioactivity data. And at the end of the day, again, to cut a long story short, what we have is 25 different ways, so 25 different descriptors associated to small molecules that tell us not only about their chemical structure, which is what we have been doing for the last 30 years, but also gives us information about their targets, the network perturbation, the effects that we can capture on cell-based assays, and also clinical information. And these descriptors are orthogonal, which is exactly what we wanted. They're not redundant, right? So just one example here. So these are different drugs. Each row represents a descriptor, a drug. If you just look at these few drugs that are related to ophthalmology, you can see that at the chemical level, they're completely different, right? So there's no structure in these descriptors. But when we look at the descriptors that tell us about mechanism of action, we can see that many of these drugs are pretty similar, both in here and in here. And if we look at the effects that they trigger on the networks, we see exactly the same thing, right? So this is information that we can capture with these sort of complex bioactivity descriptors that the chemical descriptors alone do not provide. And of course, this is not just a nice idea, it's a nice idea that correlates, as I was saying at the very beginning, with the complexity of biology. So what these plots tell here is whether they're measuring whether two small molecules that are close in one bioactivity space also share similar mechanism of action or therapeutic areas. And what you can see here is that in all the cases, we do get a significant signal, right? Both when we look at areas under the curve or when we look at enrichments of clusters of molecules that are close in the space. So what's next? So I half lied to you, right? So I said that the chemical checker is constructed around 1 million small molecules for which we have experimental bioactivity information. This is indeed true, but what is not true is that we have complete bioactivity information for all of these compounds in all the different bioactivity spaces, right? So of course, we always have chemical information, but we only have targets information for about half of these compounds and only network information for about 300K. And the most extreme case is the clinical information, which of course is only available for something like 7,000 different compounds. So what Martino did in the lab is saying, okay, so can we use all the information in the chemical checker to sort of complete this matrix. So can we predict the missing signatures for these 1 million compounds? And again, to cut a long story short, all this is published as well. The answer is that yes, to a great extent, we can do that. So imagine that we have this compound for which we have always complete chemical information and some descriptors on the targets, on the networks, and some cell-based assays. What Martino did is to come up with a pretty smart way called the Siamese neural networks, so that by comparing these compounds to other compounds in the chemical checker, we can impute the missing signatures in here. So this is bioinformatics, which means that it's not perfect, right? So we are predicting here, we are imputing these signatures, but in most of the cases, our imputations are pretty good. Right? So this means that we are roughly right in 7 out of the 10 imputations that we make. And what is more important, we have associated scores to this imputation so that we can know whether when we are predicting a signature, this one is likely to be correct or is most likely to be wrong, right? And we can use this afterwards in our predictors. And this is pretty handy because now what we have is a complete space of 25 different descriptors telling us about bioactivities for 1 million compounds. And these are vector-like, which means that we can stack just one after the other and construct a big descriptor associated to each compound that will tell us about the chemistry, but also of its bioactivity at the different levels of complexity, from the targets to the clinics. And these stacked signatures, they are indeed very well to recapitulate mechanism of action and therapeutic areas, right? With pretty impressive areas under the course. And we can start seeing the value of stacking all these signatures, right? So here we're just clustering different compounds according to their main targets, and we are displaying here the chemical diversity within each cluster. So we can see, for instance, here is this cluster. These are compounds that are known to target and modulate the activity of HSP90. And you can see that some of these compounds in blue in here are pretty similar from the chemical perspective, which means that we could have done this, we could have grouped all these compounds together already 10 years ago. But some of them are pretty different, right? And they are only here because we are able to capture their bioactivities. So dissimilar chemistry, but with similar bioactivities, and we're clustering them together. And then Martino went on and said, okay, now we do have complete descriptors for these 1 million compounds. This is already enough information so that we can build real predictors. And these are a collection of deep networks that we call signaturizers, that you provide. The input is any compound of interest, and then the output is these 25 different bioactivity descriptors for these compounds. And this is pretty cool because this allows us to start mapping, characterizing completely uncharacterized compounds collection, right? So this is just one example. So this is a 2D representation, is TSNE of these multidimensional maps. And here we have different compound collections. These are approved drugs, these are human metabolites, these are bacterial metabolites, these are compounds from plant extracts and compounds from food extracts, right? And you can see, for instance, that using these global signatures, the area of this bioactivity space that is occupied by approved drugs is quite different to the one that we have human metabolites, and radically different to the ones that we have in bacterial, yeast, or plant extract, right? And then, of course, you can always zoom in and then start exploring these uncharacterized compound collections, saying, for instance, that the metabolites that you can find in the cerebrospinal fluid and the placenta are pretty similar between them, occupying a very concrete region of the space, whereas the metabolites that you find in blood or urine are much more spread throughout the whole chemical space that we find in the human metabolites. And we can use these to characterize, as I said, the different collections. So this is just one example of a collaboration that we did with Medina a couple of weeks ago. So these guys have characterized something like 2,000 different compounds from their huge collections of natural products extracted from bacteria, from yeast, from fungi, etc. And they wanted to compare it to different well-established compound collections, in this case, again, metabolites, I mean bacterial metabolites, plant extras, or the more classical synthetic libraries, right? So if you just compare the chemistry, you can see that the overlap between these extracts and the others, the compounds, is pretty small. But when you use these signaturizers and then you start grouping, in this case only chemical descriptors, what you can see is that indeed, as expected, the synthetic libraries occupy a very well-defined region of the space, and the natural product libraries occupy a complementary region. But what is important to them is to show that only these few compounds that they have already isolated and characterized are a very well representation, very good representation of all the much broader diversity of other larger compound collections. Right? And this is something, again, that you can do by stacking the different types of descriptors that we are being collecting here. So this is just another example of the value of it. So these are a few compounds for which we know their mechanism of action. And this is what happens when you try to cluster, when you try to group these compounds using only chemical information. In this case, it's simple Morgan fingerprints, right? So you can see that in some cases, these are antibiotics. Of course, these guys are much bigger and distinct from the other compounds, so they cluster together. But the rest of compounds are sort of scattered throughout the whole space. When instead of just chemical information, you use our bioactivity descriptors, you can see that the clustering is a lot more robust, and indeed we're able to group together all those drugs that have similar mechanism of action. Again, bioinformatics, does it work always? No, not always. So we can do the same exercise, not only for mechanism of action, but for targets, for therapeutic indications, disease indications, etc. And basically what this plot shows in here is that whenever you see a blue dot, it means that using bioactivity descriptors brings some advantage in the classification. When you see red dots, it means that the chemistry alone works best. And when you see white dots, it means that it's so-so. But what you can see is that overall there's more blue than red in here. So in general, our bioactivity descriptors provide an added value to these compound classifications. And what we have seen as well, I mean, of course, at the end, as I said at the beginning, what we want is to introduce this description, this capturing of complexity for chemoinformatics for drug discovery tasks. And what we're doing here is building predictors of different complexity using only the classical chemical descriptors and our bioactivity descriptors, right? And you can see that for, let's say, easy tasks when the biological complexity is not much needed, like the capacity to inhibit the HIV protease or to bind beta secretase, our bioactivity descriptors in blue, they do roughly the same. They achieve the same performance than just using the chemistry alone. But the more complex the task is, like when we go to clinical toxicology or to cell toxicity or side effects, you can see that indeed using these bioactivity descriptors represent an added value here. And we can, our hope is that we are really capturing some of this clinical complexity in these descriptors that then help predicting whether any compound is going to be toxic in the clinics or in cell-based assays, etc. So to wrap up this part of the story, what we have is 25 different types of descriptors associated to small molecules that give us information not only of their chemistry, but of their bioactivities. This is one side of the equation, the chemistry. How about the other side, which is biology, right? So here the picture is even more complex, right? Because in chemistry, at the end of the day, what we have is one small molecule, and this is what we want to describe by using different types of information. in biology, we don't have only one entity. We have many different biological entities, from genes or proteins to tissues, cells, diseases, treatments, symptoms, etc. And each one of these different biological entities or biomedical entities have a kind of a semi-optimal way of transforming the information in the form of these vector-like descriptors. But we wanted to unify everything into a single framework. And this is what we did. This is the work of Adrià Fernandez in the lab. He was a PhD student who finished a year ago in the group. And then basically the first step here was to gather as much biological information as we could to build this gigantic knowledge graph, right? So what Adrià did here was to merge information for more than 200 different databases. And then we structured it around 12 different biomedical entities: genes, cells, tissues, diseases, compounds, etc. And then we also established almost 70 different relationships between these different nodes in the network, right? So a given gene is mutated in a cell, or is downregulated in a given tissue, or is associated to a disease, right? So this is what a knowledge graph is. Knowledge graph is a gigantic network that allows different types of nodes and different types of relationships between these nodes. Right? So this is huge. Overall, the biotech, as we call it, contains information on more than 500,000 nodes and more than 30 million relationships between these nodes. This is unique information, of course, right? But then when you try to compute this is a gigantic matrix and is very, very inefficient. So what we're doing here, we're playing the same trick that we applied for the chemical checker, and we're using a technique that is called network embedding that basically we borrowed from natural language processing algorithms. And basically what this does is that I dimensionality reduction, but this network embedding is, if correctly performed, is able to maintain the local properties, so the local distances between these elements, but also the global properties of the network, right? So what we did here, again, similar to what we did for processing the bioactivity data associated to small molecules, is try to come up with descriptors that tell us different properties of these different biological entities. This is just one example of how we use it. Again, this was published last year. So imagine that we want to describe compounds according to their relation to certain genes or association to a diseases. So what we do is we go to this gigantic knowledge graph, and then we build a specific network that connects compounds with genes with diseases. And then what we do is we set some random workers to explore this network, and all these random trajectories is what we use as if it was the corpus of text information to build these descriptors. Then we play with these embedding techniques. And at the end of the day, what we have again is a descriptor in the form of vector that tells us about compounds and about diseases, but not any type of information. We don't have chemical information here in the compounds. We only have information associated to the proteins that they target and the diseases that they treat. And again, to cut a long story short, what we ended up here is having more than 1,000 different, what we call context-dependent biological descriptors, which are similar to the 25 bioactivity descriptors to the molecules, but in this case, it's more than 1,000. Why do we call them context-dependent? Because basically these are different ways of describing these 12 biological entities. So imagine a protein, for instance. You can describe it by its name, its amino acid sequence, but you can also describe it according to whether this protein is expressed in a certain tissue or cell line, or whether it interacts with certain pathways, or whether this is associated to a disease or targeted by a specific small molecule, right? So these are different ways of describing the same entity, and they capture different things. And this is exactly what we're doing here. So just a couple of examples. So imagine two genes here. So these two genes have exactly the same function, which means that their descriptors at the function level are going to be almost identical. They localize in the same subcellular locations in the cell, and they belong to the same biological processes. But one of the genes is expressed in one tissue and the other is expressed in another tissue. So this means that three of these descriptors are going to be almost identical, but the fourth descriptor is going to tell us that these two genes are not the same, right? Or just another example based on cell lines, which are pretty relevant to the open screens, I believe, right? So two cell lines, they harbor the same mutations, they have the similar transcriptional behavior, but when we go from transcripts to protein abundance, we can see that they are already different, right? And then this difference in the protein abundance, in the clearance of these proteins, translate into a very different sensitivity in front of drugs. So again, in this case, the descriptors related to these cell lines, two of them are going to be almost identical, two of them are going to be different. Good. So we have now vector-like descriptors, so something that computers understand for the chemistry and for the biology. So now it's time to start blending these two descriptors to come up with a way of finding modulators at high level. What do I mean by this? So you can start from your favorite biological signature, either coming off your lab or from the biotech, and then you can look for compounds that are able to mimic or revert a particular signature. But we don't have to go, in this case, through the chemistry or through the targets. So we can go at a much higher level and look for compounds that are able to mimic or revert signatures that are related to gene expression or disease comorbidities or even disease symptoms, right? A few examples of this. So as you can imagine, most of the data that populated both the biotech and the chemical checker come from cancer cell lines. And we wanted to make sure that our descriptors could be extrapolated to other systems that the chemical checker had never seen, right? So what we did is to use these SY5Y cells that are neuron-like, and then we artificially introduced in the lab different mutations that are related to the familiar Alzheimer's disease, right? So that we now have neuron-like, wild-type neuron-like cells and 80 neuron-like cells. And then what we did was to run just differential gene expression experiments, and then we know the changes that the 80 neurons have with respect to the wild-type neurons. And we went to the chemical checker and said, look, do we have any compound that we think can revert these transcriptional signatures? So in other words, to reconvert these sick neurons into healthy ones? And to cut a long story short, as you can imagine, the answer is yes, because otherwise I wouldn't be telling the story. And what we found is different compounds. Some of them are approved drugs, some of them are just experimental compounds that to some extent are able to revert these transcriptional signatures related to Alzheimer's disease. We are able to overexpress the genes that are underexpressed in 80 neurons and to downregulate the ones that are overexpressed. And these genes are not just random. Indeed, they are the ones that are closely related to the disease. In this case, BIM is involved in vesicle endocytosis in the synaptic membranes. RIM2 is the glutamate receptor, again in the synaptic membrane. So these are very relevant genes. So good, we're happy. We're able to somehow capture this cellular complexity. Can we go higher? And basically, in this case, what we did was to use 3 different AD mouse models. And then, of course, we ran some behavior tests. And then we took the hippocampus of these animals, which is where Alzheimer's disease starts. And then we run full proteomics and transcriptomics analysis of these hippocampus. And to cut a long story short, we ran some classical bioinformatics analysis in here. And what we saw is that there were some modules, some groups of genes that did not change in expression in healthy aging. right? So when healthy mice are growing, 3 months, 6 months, 9 months, this module remains stable. But in AD mice, in the sick mice, these modules were shooting up. And this is something that was happening both at the RNA level, but also at the protein level. So this module 2 was a perfect candidate for trying to identify these compounds able to revert these signatures. This is exactly what we did in the checker, but in this case, these are not cell cultures, these are mice, which means that we cannot just use funny compounds. I mean, we have to use approved drugs in this case, right? And what we did is let the mice grow for 5 months, then we treated them for 4 weeks, and then we checked whether we could see any behavior change and also biochemical changes. So we identified something like 40 different compounds, but then what we realized is that most of them could be grouped in the non-steroid anti-inflammatory drugs or antihypertensive, right? So 2 very well-defined groups of drugs. Then what we did was to pick 6 compounds belonging to these 2 groups that had never been associated to Alzheimer's disease in any way, right? As I said, we treated the mice for 4 months, and basically what you see here is the result of the behavior test. In this case, it's a novel object recognition. I don't know whether you're familiar with it, but basically what you do, you have your cage, then you put 2 identical objects in this cage, you have the mice, they explore them, and then after some time, you remove one of the objects and you introduce a completely new object, right? So healthy mice are very curious in nature, and basically what they do is they spend most of the time exploring the new object because they remember the old object. But the AD mice, they just don't remember, and they spend roughly the same time exploring the new or the old object. So what you can see here is that for 4 out of the 6 drugs that we tested, we could recover the wild type phenotype in this novel object recognition test, right? So these are phenotypic level. So these mice were behaving like wild type. At the molecular level, what we saw is that indeed, for these 4 compounds, we were able to downregulate to some extent the overexpressed genes in AD mice. And in one case, pembutalol, that indeed is the one that worked best in the phenotype, we were also able to push up the genes that were downregulated. Other examples. So in this case, what we want to do here is to mimic the activity of biodrugs. So you know that the monoclonal antibodies are very trendy now. They're very, very powerful, but they're expensive to produce and difficult to handle. So the idea here is that if we could find a small molecules that do the same phenotypic effect that these biodrugs, that these monoclonal antibodies, this could be a good asset for the pharmaceutical companies, right? So in this case, we started by studying daclizumab. So this is an antibody that is used to avoid rejection upon organ transplantation. And we know that this antibody, what it does is to target the receptor of interleukin-2, right? So in this case, what we did is genetically, I mean, using shRNA, we blocked this IL-2 receptor. And then again, we run some differential gene expression experiments here. And then what we said is we went to the chemical checker and said, do we have any compound that is able not to revert in this case, but to do the same than genetically killing the IL-2 receptor transcriptionally? But then we also said, look, and we don't care about the target, right? So we want the same effect. We don't care at all whether you touch interleukin-2 receptor or something else. This is why we use the network like descriptors. Right? And then we use exactly the same experimental setting that was used to discover daclizumab, in this case, using peripheral blood cells. Right? And what you can see here is that we tested 19 different compounds, and for 14 out of these 19, we got a very, very significant reduction of the proliferation of interleukin-2 dependent cells. Right? I'm going to walk you through one of these experiments. In this case, it's for compound 20, it's this one here, uncharacterized compound. So basically what you see here is that when you have the cells without interleukin-2, the cells do not grow. When you add interleukin-2, cells start proliferating, and when you add the teclisumab, you stop the proliferation, right? When you add our compound number 20, you stop also the proliferation, even to a greater extent than the teclisumab itself, in a very nice dose-dependent manner. Good. But as I said, we don't care about the targets here, right? So what we know is that downstream of interleukin-2 receptor is STAT5 that gets phosphorylated. So this is exactly what we're checking. So when there's no interleukin-2, there's no phosphorylation. When we add interleukin-2, STAT5 gets phosphorylated, and the teclisumab blocks this phosphorylation. But with our compound 20, the phosphorylation of STAT5 is completely unaltered, which means that we're able to achieve the same effect. So we're stopping specific IL-2 dependent proliferation of cell growth, but without touching STAT5 or this pathway. So we're doing it through a completely different mechanism of action. And this is just to show you that this is not a nuclear bomb, that indeed the proliferation that we are stopping is IL-2 dependent. I'm going to skip this for the sake of time. And then I'm just going to give you one real example. So I don't want that you end up thinking that this is pure magic and that always works and we're able to cure everything. So this is just one ongoing collaboration with a group of Eduard Batlle here at the IRB Barcelona. So these guys work on colorectal cancer. And what they want to do is to stop the Wnt pathway, but very, very downstream of this pathway, so below the beta-cathepsin. So what these guys have is 10 different colorectal cancer cell lines that they have inhibited this pathway below beta-cathepsin genetically. And what we want to see is whether we can find, again, chemical modulators able to do exactly the same thing. And what they have, again, is transcriptional data on these genetically modified CRC cell lines. Then what we did in this case is to use all the compounds in the L1000 repository. We also used everything that we have in the chemical checker and in the IRB library in here. And overall, we could identify 2,600 compounds that we thought might have this potential. Then we filtered them by applicability, is our sort of reliability score, distances, etc. And finally, we ended up with 357 different compounds that we thought would achieve the desired effect. Of course, what we want to measure here is the added value of using the bioactivity descriptors and the biotech descriptors. So basically what we did is we also picked 320 random compounds from the IRB library. And these are the results here. So in a nutshell, 90 out of the 357 compounds that we selected are positive, right? And only 4 of the randomly selected. Then again, this is not perfect. So only 90 out of the 350 that we picked show the desired effect. But again, it's not magic, but it's a huge enrichment, 25-fold enrichment over the random. So you can just play these Mickey Mouse, easy bioinformatics, chemoinformatics approaches and get a huge enrichment in your screens. And this is kind of my wrap-up slide. So I have presented today the chemical checker, which is this repository that contains experimental bioactivity information for about 1 million small molecule. We have developed these bioactivity fingerprints that allow us to extend the similarity principle from chemistry to other levels of complexity. And I also showed you this signaturizer so that we can infer this bioactivity signature to any molecule of interest. From the biological perspective, I showed you the biotech, which is this resource that collects and harmonizes in a qualitative form most known biology. And we have also derived these biological signatures to integrate in a format that is suitable for modern machine learning. And what we're doing, and I showed you several examples on biotrucks, Alzheimer's disease, is blending these bioactivity descriptors for small molecules, biological descriptors for complex phenomenon, and trying to find a common ground in here. And I always say that this is pretty similar to the parable of the Indian blind men that go into the jungle. I don't know whether you're familiar with it, but the story goes basically that is these 5 or 6 Indian blind men that go into the jungle, and then they bump into an elephant, right? They're blind, so they cannot see the elephant. So this guy here says, look, I found a rope. I say, what do you mean a rope? It's a wall because I cannot pass. Or no, it's a tree because I can hold the trunk in here. So the idea is that each one of these guys have partial information on the system, right? So if we were able to integrate all this partial information, maybe we could come up with a better idea of reality, that in this case is that they bumped into an elephant. So it's the same for us. We have 25 different ways of describing small molecules. We have 1,000+ ways of describing biological entities. If we can combine all of them in a meaningful way, maybe we will get a better idea, a better representation of the biological reality and the small molecules that we can use to tinker with this reality. And finally, as you can imagine, I did very little of this work myself, apart from presenting it and writing it up. So this is my group, and this is just to show you that it's a very multidisciplinary group. So in here, we have computer scientists, we have, now is a double degree in physics and chemistry, we have biotechnologists, we have geneticists, we have biologists, we have chemists, we have pharmaceuticals, and I'm a biochemist. So we have a little bit of everything from technology to pure biology, which is what we need to come up with these descriptors and then test the experiments in the lab. And I'll stop here and I'll be happy to take questions. Thank you very much.
Many thanks, Patrick, for this interesting presentation. Me, myself, I really enjoyed the specific experimental layouts. So yeah, I think we have already a raised hand from Ruben. Ruben, maybe you can pose your question directly to Patrick. Ruben, can you hear us?
He's mute. I can see it. Okay. Yep, we can hear you.
I cannot hear him though.
I'm fine, I can hear him. You're mute again, Ruben.
I cannot hear Ruben for some reason.
Okay, I think I got the question. So the question, let me repeat it, is basically whether we have used our descriptors to compare the huge libraries that you have in OpenScreens and whether we can make predictions out of it, right? So the short question, or the short answer, is that yes, we have done it, but to some extent and only for a different purpose, right? So basically, we have our drug screening platform here at the IRB Barcelona that, in fact, I know that they want to become, or they would be happy to join the OpenScreens initiative in here. And basically, we were increasing the number of compounds that we have in this library, right? But we want a library to be as unique as possible while maintaining the chemical diversity. So what we have done in here is to explore different vendors, different collections, including the ones in OpenScreens. And then from these several millions of compounds, we have selected 200,000 compounds, plus some RNA binders and covalent binders and fragments, etc., that are able to cover the whole chemical and bioactive space, but that they are different to the ones that you are offering. so that our library is going to be slightly different to the ones that you have. And if we discover something in here, I mean, we can sort of be sure that we are the only ones working on it and not the whole Europe. So this is how we have looked at your libraries and annotated them. Having said that, if you want to have the annotations, or I forgot to say, but absolutely everything that we do is open source in here and open science. So you can use our signaturizers, you can build descriptors for all your molecules, or if you prefer, we can run it for you, and even the annotations. And if you think this might be useful to OpenScreens, I'll be happy to share it.
Thank you, Patrick, and thank you, Ruben, for the question. Another question coming from our participants: what are the main challenges in developing and applying systems pharmacology models?
Main challenges, there are very many. So there's not, I mean, basically, basically our idea here, so what triggered everything is this notion that we want to go beyond target-based approaches, right? So we know that the target-based approaches are very good for building me-toos, so to kind of fine-tune drugs that already work, not to find cures for diseases for which we don't have compounds, right? And then one of the reasons in here, as I said, is this biological complexity. So you can find a pretty good target in vitro, but then when you put this compound in vivo, is not going to work. So our idea here is to include a little bit, as much as we can, of this biological complexity from the very beginning, right?
Yeah.
So the part that we're doing now in the lab that I didn't present is, as many other people, we're playing with generative models. So starting from your huge libraries, we're kind of fine-tuning, developing computationally new molecules that are kind of biased towards having less toxicological effects, etc.
Right.
This is what we're doing. This is a huge challenge. I don't see how we're going to solve it in the next couple of years, but hopefully in 5 or 6, this should be a reality.
Great, thanks, Patrick. Another question, the development of detailed models in quantitative systems pharmacology, QSP, requires a significant amount of biological data, and models often require knowledge of specific mechanisms. How we can overcome this? Can you comment also on Boolean modeling techniques? For those who don't know, in computer science, the Boolean is a data type that has one of 2 possible values, true or false. Yeah.
So these are other. So first, I mean, these are perfectly valid approaches. This is not what we do in the lab. Why not? Because they usually are restricted to pretty well confined models, right? So as the attendant is saying, so you need very detailed information. So this information is not the one that we have on very complex systems. We just have what we call omics information, right? So we're trying to go away from these models that work very well when you are studying just one pathway, right? But not in general terms. So, but they're perfectly valid. And then I forgot the first part of the question, sorry, before the Boolean models.
Okay, the first part was about that QSP requires a significant amount of biological data, yeah.
Yeah, so here, I mean, we can never go, unfortunately, we can never go quantitative, right? But what we can do, I mean, as I said at the very beginning of the talk, I mean, our first thing, we're trying to compress as much bioactivity information and as much biological information in the form of one single descriptor, right? So this means that we are pushing the effects on 80 different cell lines in the form of one descriptor. Yeah. Then when we go to specific projects working with pharma or here in the lab, I mean, of course, you don't want that. I mean, you don't need these 80 different cell types because 70 of them are not relevant to your system. Then what we can do is we can build less general but more focused descriptors tailored for a particular problem, right? Then again, this is not quantitative yet, yeah. but at least it's more focused, and then you can sort of use this information to go and develop what we call these SAR models, that are signature activity relationship.
Yeah, great. Thanks, Patrick. I think we have time for one more question. Can you comment on the one drug multi-target pathway network systems pharmacology drug versus the one drug, one target approach?
So what I can say, if I understand correctly the question, what I can say is that for most complex diseases, I mean, Mendelian diseases are something different, right? So you have one problem, we fix that problem, that's easier. For most complex diseases, I mean, just touching one target is not going to achieve the desired effect, because of the robustness of the complex biological systems, right? So you block whatever cell proliferation in here, but then there's a squillion different alternative routes that the cell is going to find, right? So this is one problem. Problem number two, if you only touch one target, basically what is going to happen is that unless you can fix it once and forever, which is very difficult, is that the resistant mechanism will appear, right? So either by inactivating the drug or by mutating the target or through finding alternative routes or things like that. So if you go for general blockades using the whole pathways, whole networks, then I mean, my hope is that the blockade is going to be more effective and more difficult to overcome for the cell if we're talking about cancer, that we want to kill the cells, not cure them, right? But that's why I said, I mean, this is I mean, you can find it in Nature Reviews Drug Discovery every year, right? So again, first-in-class drugs are still mostly found by phenotypic screens. I mean, not by target-based approaches. I mean, me too's perfect, but not first-in-class, and this is what we're after. I mean, trying to capture the kind of limited complexity of these phenotypic screens into a sort of descriptor so that we can run them in the computers.
Great. At this point, Patrick, I would like to thank you very much for this interesting discussion that we had, and I hope in the near future we will have the opportunity to talk more about this subject, as it's the future of biochem in any way. So thank you very much for being here for us today.
My pleasure. Thank you.
Thank you.
Ciao.
So before we close our webinar, I would like to remind you that you can find us and you can learn more about EU-OPENSCREEN and our training activities in our website, www.euopenscreen.eu, or you can contact us by email in our email address. You can also subscribe to our newsletter in order to receive news, and you can always check about our training opportunities in our website in the news tab. Thank you very much for being here today, and hope to see you soon in our future webinars. Thank you very much.